So good morning, everyone, and welcome to the M-LEEAD's Summer Omics and Data Science Symposium. So everyone on Zoom can hear me this morning. So this is hosted by the Panomics and Data Science core of the M-LEEAD Center. That M-LEEAD Center being the Michigan Center for Life Stage Environmental Exposures and Disease. I am Maureen Sarter, lead of the Panomics and Data Science, or PODS core, in the M-LEEAD Center. So probably most of us are affiliated with the center in some way, but special welcome to anyone here or on Zoom today who are not affiliated with our center. Welcome. So probably most all of us are well aware that environmental exposures can increase the risk of a variety of diseases, right, at different life stage time points, and that they can do so through a variety of complex combinations of different types of molecular mechanisms and in specific tissues and cell types. And so these changes can involve changes in epigenetics, DNA sequences, RNA expression and splicing, post-translational protein modifications, metabolites, microbiome, like you name it. Environmental exposures can do one or multiple of these things. And so that's where we come in. Any of these, and furthermore, any of these different types of molecular changes or signatures from them can potentially be used to predict phenotypes or clinical outcomes. So we also help people with those predictions, and the resulting data can be used to develop software tools for environmental health science community. So the main goal of our PODS, the Panomics and Data Science Core, is to help M-LEEAD science members and the overall environmental science, health science community accomplish these tasks, right? So first, we aim to provide comprehensive support through the entire process of an omics experiment or potentially software tool development from the initial study design through to publication. We aim to help we aim to develop some of our own novel data science methods and tools to help meet the needs of environmental health scientists in the M-LEEAD Center and beyond. And so today, from our outstanding lineup of speakers, you will hear examples of all of these goals of the PODS core. So you'll hear both from bioinformaticians who have developed novel methods and have analyzed complex omics experiments, as well as hearing from M-LEEAD members who have benefited from our bioinformatics services and other services in the PODS core. So I will introduce our first two speakers this morning, who have both been very active M-LEEAD Center members for several years. And then my colleague, Dr. Jackie Goodrich, many of you know, will introduce the remaining four speakers who are all exceptional early career faculty in the M-LEEAD Center. So to get started, our first speaker is Justin Colacino, an associate professor in environmental health sciences and nutritional sciences. Justin received his MPH from University of Texas, MA in statistics and PhD in environmental health sciences from University of Michigan in 2014. And I had the pleasure of working with him on his dissertation committee. And after that as well. And so main goal of Justin's lab is to characterize the susceptibility of stem cells to environmental stresses and help better understand the links between these stresses and the increased risks of various diseases, complex diseases such as cancer. And so to do this, as you might imagine, requires understanding cell type and tissue specific changes, which today is really optimally done using various single cell and spatial omics techniques. And so Justin has become somewhat of an expert in these things, especially related to environmental health sciences, and has been involved in University of Michigan's efforts pretty much from day one, right, with our single cell group and spatial analysis group. So thank you, Justin, for being here today, and I will turn it over to you now.
Great. Thanks, Maureen, for the very kind introduction. Do I just like move forward? Wow.
Right on schedule.
I'm starting a stopwatch because this is a whirlwind talk. Okay, yeah, thanks so much, Maureen. Thanks to Jackie for the invitation to come talk today. So this is not going to be a research talk per se. This is going to be more like a resources talk that might be available to you if you're interested in trying to integrate some of these single cell and spatial omics methods into your own environmental sciences work. I'll go through some different methods. I'll give some like tips and tricks, some things that we've learned using these methods in our lab. And I'll give a couple of vignettes where we've used these different approaches in EHS type studies. And so, like Maureen was saying, we've been interested in this area for a long time, but you might be wondering, why would I want to incorporate single cell studies into my EHS work, right? And so all of us are probably aware that if we're looking at an individual tissue, that tissue is made up of different cell types. And surprise, surprise, those different cell types have different molecular profiles that define their structure and function, right? And so anytime we are measuring a bulk tissue, we're looking at an average of the molecular profile of all of those different cell types, right? So this cartoon here is just depicting this on a simple basis. So let's say we have a tissue that's made up of 3 different cell types, and we're looking at the molecular profiles of these different cell types. Maybe cell type 1 has very high DNA methylation. Maybe cell type 2 has medium DNA methylation. Maybe cell type 3 doesn't. If we're capturing the average of all of these, we're just getting like an average level of this. So that looks across the average of all these different cell types. But in the case of an environmental exposure, what if that average changes, right? That could be the result of a few different things. And so this is a very nice figure made by Kyle Campbell and Kelly Bukowski. But yeah, we could have a direct effect, where the chemical exposure is causing an increase in methylation or an increase in the molecular marker across all cell types, right? That's what's being depicted here. We could have an individual cell type that's vulnerable, right? Maybe the effect is only in a single cell type, and that's what we're seeing here, right? Or we might have an option where maybe this exposure was impacting differentiation, caused the differentiation trajectory to change, and led to different proportions of the cells. This like cell type heterogeneity issue. All of these would lead to the same outcome if we were looking at bulk data, but those have very different meanings from an environmental health standpoint, right? So this like gives us an idea about why single cells might be important. Sort of in parallel, right, there's been this huge explosion of all these new technologies that Maureen was alluding to, that are now these new spatial methods for giving us new and exciting ways to measure tissue or cellular structure in organization, right? So thinking about cell shape and size, right? The organization of various organelles in the cell, right? These are very much defining tissue or cellular function, but then we can also look at a tissue level, right? Where we have individual cells, we have cells and their neighbors, we have little neighborhoods, we have tissue microenvironments. We know that there's a lot of important regulation that's happening, and then dysregulation in diseases. So there's been a huge push in cancer, in Alzheimer's disease, many other diseases, and now start to use these spatial methods to really try to understand, the drivers of these diseases at really an organizational level. And I think now we are primed in environmental health sciences to start to apply these types of methods to really trying to understand how environmental stressors impact disease. So you might be wondering, like, what might be available to me to do these types of studies? So if you want to jump into the single cell type of analyses, there's a couple different buckets of analyses that you can try to do, right? There's well-based methods. This is where you're pipetting cells into an individual well, you're lysing those cells, and then you are doing some sort of molecular profiling on those individual cells. There's droplet-based methods, which are by far the more popular based methods these days. This is where we are using tools like the 10x genomics chromium, which I'll talk a little bit more about, to profile many, many, many cells. Right, so these are very powerful techniques, but now there's this huge variety of spatial methods. I think as I'm talking now, more and more spatial methods will come out on the market, like during this lecture, right? It's like moving so fast. But I'm going to group these into various flavors of assays. So I'm going to talk about some assays that measure sort of like the unsung hero of omics, what I call morphomics, right? Where we're looking at cellular morphology using high content imaging and microscopy. There's antibody-based methods if you're interested in protein expression across a tissue at sort of like lower plexity up to maybe 100 proteins. There's some powerful tools that we have here on campus to do this. There's sequencing-based methods, right, where we are doing sequencing across a tissue. So you get spatial resolution of, say, like RNA expression patterns across a tissue, very powerful. And then there's in situ hybridization-based methods, where we are doing, say, like RNA in situ hybridization at very high plexity to really get an in-depth idea of, say, like RNA expression across the tissue. So there's a lot of like really cool and powerful techniques that have hit the market to really start to allow us to dig in on the specific biology. And so today I'm going to really be focusing on these droplet-based methods, high content imaging, and they give you like some ideas about these. These have like just hit the market, right? So these are like brand new. I'll give you an idea about what's going on here. So let's talk a little bit about single cell profiling. And so if you're doing single cell profiling, you need to like have a single cell suspension, ultimately, right? That's probably not too much of a surprise. So if you're dealing with a solid tissue, so we do like a lot of breast cancer work, you need to take that tissue, that solid tissue, and dissociate it somehow into single cells. If you study blood, you're lucky, right? Your tissue of interest is already in single cells. So this is like already a place we need to think about, where now we already have a pause point when we're designing one of these experiments, right? You have to think about how are you going to do your tissue dissociation, right? There's a lot of considerations you have to think about when you do this. So probably bullet point number one isn't surprising, right? Different tissues are going to require different dissociation protocols. I see Claudia nodding because Claudia knows this very well. We've like talked about this a lot. You know, the tissue dissociation protocol that you're using can also significantly impact your results, right? So there might be multiple tissue dissociation protocols for a specific tissue. One might enrich for cell type A, another one might enrich for cell type B. You really want to know what's going on before you get in. Usually what we're doing for this is we're combining physical dissociation strategies. So this can be like you with scalpels mincing. You can like grind things up. With enzymatic dissociation strategies, things like collagenase and hyaluronidase and disphase, right? And I would recommend, if you're like into this, explore some commercially automated solutions that do this. So like in our lab, we have a Miltenyi GentleMax, which can do 8 samples at once. It makes things like super consistent via a reproducible protocol. So you want to think about some specific quality concern, some specific things to think about for quality control. I talked about how digestion methods might differentially enrich for specific cell types. If you are doing viable single cells, you need to make sure that they remain viable through the process. That can be quite challenging, depending on what tissue you're using. Some tissues just like don't love being dissociated into a single cell suspension. And then I would highly recommend that before you move on to doing an extremely expensive experiment, sort of like validate that your tissue dissociation is giving you what you expect. You know, look at the cells under the microscope, maybe do some flow cytometry with a no marker panel, or maybe just even do some basic qPCR to kind of like give you an idea about that you're getting into what you think that you're doing. And so by far the most common tool that's available and used on campus is the 10x Genomics Chromium, which we have at the Advanced Genomics core. This is a droplet-based sequencer, which takes advantage of 10x's like cute little microfluidic device. And so what's happening is our cells are flowing in as a single cell suspension, and then a single bead suspension is also flowing in. So 10x has these what they call bar-coded gel beads. So each of these beads is specifically oligo-tagged. We're making a little reaction vessel and like a little droplet. You lyse the cells, you generate cDNA, and because of that barcode tag, that barcode then gets integrated into all of the cDNA. So then we can sequence all of these RNAs, or well, now cDNAs, look for those barcode tags and figure out which RNAs came from which cell. So like pretty cool technology, right? There's some things that you can think about, including some like new technology that just came on the market a couple months ago, that you should be aware of. So if you're starting with viable tissue and you want viable cells, this is kind of like the example I just talked about. I don't want to do physical and enzymatic digestion to collect viable cells. This needs freshly collected tissue, so it's more amenable to in vitro studies, in vivo studies, or if you have like a direct line with the hospital, maybe one of those studies. Alternatively, you can do nuclei isolation. So you can start here with pressure frozen tissue, potentially even fixed tissue. You dissociate the tissue, you capture the nuclei, which is being depicted here. And then you can do what's called single nuclei sequencing. So you're capturing a little bit different data, but you can leverage all of those toxicology tissues that you have in the freezer, right? So that's like really powerful. And now there's a new method that came out from 10x that we are currently trying. I can't quite endorse it yet. We have like some breast samples and some colon samples at the core right now. But the potential is that you can take fixed or frozen tissues now, and then if the frozen tissue is frozen, you fix it, and then you dissociate that fixed tissue into a non-viable but fixed single cell suspension. And you can profile that. And so 10x has a new method that came out called the CellPlex method that does that. And this is very applicable to toxicology or environmental epidemiology type studies, which is why I'm highlighting it here. And so as you're thinking about what type of data can I generate, right? There's a whole mess of data that you can do. So the classic example is whole transcriptome analyses, 3' profiling. The gene expression, this flex panel is probe-based, but still whole transcriptome. If you're interested in epigenomics, there's a TAP-seq at the single cell level, which is really cool, right? Looking at chromatin conformation at the single cell level. There's even multi-omic approaches where you can simultaneously look at RNA and chromatin conformation in the cell. And then if you are an immune fan, there's a whole suite of immune profiling. You can look at cell surface proteins, right? Using antibodies that are oligotagged. You can look at full-length T cell receptor, B cell receptor sequencing, and then you can really screen for antigen specificity. So there's like a whole menu of options that are available that kind of like add on to this Chromium kit. And so let me give you a quick example of how we've used this in the lab, where we were looking at the single cell effects of lead exposure on the hippocampus in mice using this Chromium method. And so this was an experiment that's part of the TaRGET2 Consortium, which I'm pretty sure we'll hear about a little bit more today. But in general, what was happening is we were doing an early life exposure to lead in mice. We stopped lead exposure, and then in the offspring later in life, we sacrificed them, and then we wanted to see like what changes had happened in the brain. So we looked in their hippocampuses, we took those hippocampuses, we did that dissociation protocol using the GentleMax in my lab, right? So we took the hippocampuses, we dissociated them to a viable single cell suspension, and we did that Chromium experiment. And so we can then start to see like, hey, what does this look like? Probably some of y'all have seen this. This is called T-SNE plot. There's like UMAP plots. There's a bunch of these different dimension reduction plots, but each point on this plot is a cell. And if points are closer together, those cells tend to be more related. If they're more dispersed, they tend to be less related. And then you can do what's called clustering, right? You look at the expression profiles and you see like, okay, these cells have a similar expression pattern. And then you can see like which of these clusters might correspond to known cell types in the brain. So you'd be like, okay, well, we see that there's some cells that express markers of microglia, some cells that express markers of oligodendrocytes, endothelial cells, neurons, et cetera, et cetera. And so, but this was like a lead exposure question, right? We're like, does lead cause changes to the brain, right? And so our first question was like, does it potentially change the proportion of different cell types in the brain? Kind of like getting back to that first slide I had. Or like, actually, indeed, it seems to. So what we saw is that lead exposure increased the proportion of oligodendrocytes and oligodendrocyte progenitor cells. For those of you who might be sort of like attuned to this, you might be like, this is a very small number of neurons. That highlights the challenge with some of these techniques where our dissociation technique did not enrich for neurons. So this just like gives us an idea about like, hey, we're doing a good job of capturing the glial cells. This method didn't do a great job capturing the neurons. For instance, it's just like gives you like a real life example of how this works in the lab. But ultimately what we're seeing is that glial cells here can be vulnerable. We can then do like a pseudo bulk profiling where we're looking at changes in gene expression from all of the control cells versus all of the lead cells. That's what's being depicted here. We can see that there's like a small number of changes that happen. What we did, someone in Kelly Bokulski's lab was working on this with, did an enrichment test. We did see that overall we saw changes enriched in psychiatric disorder or GWAS associated genes, kind of interesting. But I think what was even more interesting is that if we looked at gene expression changes, we saw that they were extremely cluster specific, right? So this is like looking at each individual cluster now, we see that by far the majority of changes are unique to each cluster. That's what's being depicted here in this upset plot. So this really gives us credence that like, man, if we're going to be doing these experiments, we should probably be capturing this data in a cell type specific way. And so we had done that in the most expensive way possible, which was fine at the time. But there are new ways that have come out to do so where you can save money, which I think is really nice, as a PI. And so one of the cool methods that's come out is called the CellPlex method. And so this is from 10x. And CellPlex allows us now to multiplex multiple samples into a single library prep, which can save, a pretty substantial chunk of money. And so here what it did, we're starting with individual samples, and then CellPlex comes in with these barcoded lipids, where you tag now each sample with its own barcode. You then pool these guys together, and then you can go through the normal prep. And then when you're sequencing your RNA, you can figure out, hey, this RNA came from this sample, and then, hey, this RNA came from this cell. So this really allows you to increase the throughput. You can now pool 12 samples together instead of doing just one sample at a time. So now we're starting to get into the realm of public health, right? Another tool that I want you to be aware of that we've had good success with is what we call genetic multiplexing. So here we can take advantage of if the cells that are going into sequencing come from individuals with different genetic backgrounds, we can then sequence things, look at SNPs in the sequencing data, and then figure out which cell came from what person. So this is extremely powerful, you can imagine. Right? So we come back, we call the genetic polymorphisms from the sequencing data. Of course, you need to know each individual person's genetics, right? But then based on that, you can assign those cells to those individual people. There's a lot of unique power here. So maybe some of y'all are familiar, but as we're doing these single cell profiling methods, there's a potential that 2 cells might get captured in a droplet rather than just 1. This is cool because you can see this droplet was actually 2 people, 2 people's genotypes. You can toss that out. So there's actually a lot of added power here. And there's some cool bioinformatics approaches that do this, and included DMUX that was developed here at University of Michigan. So we've been able to do this where we combine genetic multiplexing plus the CellPlex method to really increase the throughput of these single cell experiments, right? So we can test multiple chemicals, multiple doses across multiple people. Gives you a really good opportunity to do dose response studies. You can do comparative toxicology studies, and then also get an idea about inter-individual response to chemicals, right? So this is just giving y'all an idea about how this goes. So I'm going to shift on the back half and talk a little bit about spatial methods now in EHS. I'm going to talk about high content imaging as an unsung hero of omics and EHS. Then I'm going to talk about 2 different types of sequencing, more like traditional omics profiling across a tissue. We'll do the sequencing-based approaches, and then we'll do the in situ hybridization-based approaches. So let's talk high content imaging. And so for those of y'all who are not aware, high content imaging is really leveraging automated microscopy and computer vision to tell how chemicals or drugs or genetic perturbations impact cells. And so what a general protocol of this looks like is we take our cells, we're going to do some sort of perturbation to these cells. In our lab, we're doing toxicology, obviously. So we'll do some sort of chemical perturbation of the cell. We'll do these experiments in very high throughput, 384 well plates, right? Where we're testing lots of chemicals at lots of doses. We're doing automated microscopy imaging and then automated image analysis, which leads to these very cool, single cell data now that have single cell morphological profiles. Right, and this can be things like the size of easy stuff, like the size of the cell, the shape of the cell, the intensity of stains in the cell, looking at the granularity of different markers in the cell, the textures of the cell, spatial relationships between the cell. You can get a ton of data just from these images. And so what we think, and hopefully other people are thinking this too, is that this method can be super useful for identifying different concentration response relationships, and then really trying to start to probe mechanisms of action of chemicals. I'll show you an example of how this works in real life, but we can do it in a very, very high throughput manner now. So I'm going to give a streamlined example where we have a hypothesis-driven question, and then I'm going to give you a little bit more of an open-ended example. So this is data here from Dr. Morgan, who we'll be hearing from later in the afternoon. So Rachel had gone through, and she had developed this assay in the lab where she can take these SHSY 5Y neuroblastoma cells and then differentiate them into these neuron-looking cells over the course of many days. And so what Rachel then does is, before this process starts, she can then perturb it with a toxicant. In this case, she was studying lead. So you can add in lead, do some, grow these cells in culture, induces differentiation, and they have this like very nice phenotype at the end where we're like, oh yeah, we like have this like sort of like branching structures, and then like throwing off things that look like axons and dendrites. So like pretty cool. And so how this works in general with this protocol. So she did the differentiation in the presence or absence of lead. You label with antibodies in this case, or a nuclear stain. And so Rachel had a very directed hypothesis, which led to the antibodies that she chose for this experiment. We did high content imaging at a very fancy confocal microscope in our collaborator Johnny Sexton's lab, where we're capturing thousands of images. We ended up getting data on 250,000 individual cells just in this experiment, right? And then we can process images with this very nice software called Cell Profiler, where we're taking all those thousands of images and then we're doing those morphological measurements on each individual cell. And kind of like how this looks is that we go through, we find all of the nuclei that tells us where the cells live. We can then identify the outlines of each of the individual cells. Then we do this cool skeletonization where we can see like, hey, what does the neuron pattern look like? And assign it to each cell. We can look at like, how long is each neuron? How many branches are there for each cell? So this is like a very powerful and cost-effective method to get really good, high-quality data in this way. And so it's just to give you an idea about like what happens over time. Right, so here's the differentiation protocol. You can see these cells are changing, right? They're starting to throw out their neurons. And then we can look and see like, hey, what happens in the presence of lead? And you're like, oh, these like sort of intermediate doses of lead seem to be having like a very profound effect on the morphology and structure of these cells. And so Rachel then went and quantified, for example, neuronal projections and neuronal branching. And that kind of validates what we saw in that example where it's like, yeah, we can now statistically test across all of these cells. Yeah, we do see a decrease of projections and length of projections. So quite interesting, quite cool. And so that was a nice hypothesis-driven example, but you're like, what if I don't have a hypothesis? What if I want to do discovery research, right? I don't know what I'm looking for. Cell painting might be for you. And so cell painting is another high-content imaging-based approach that was developed by Ann Carpenter's lab at the Broad. And this is a very cool one. So we're still doing the same thing up front. We're starting with healthy and diseased patient cells. We're doing some sort of perturbation, right? It could either be a drug perturbation, chemical perturbation, toxicant perturbation, and then we're doing a high throughput assay. But here, cell painting, we're throwing in stains that measure each individual organelle. These aren't antibodies anymore. These are just fluorescent stains that are tagging all these individual organelles, right? So the endoplasmic reticulum, the nucleolus, the nucleus, F actin, mitochondria, cytoplasmic RNA, Golgi apparatus, and the plasma membrane. And then we're going through the same process where we're identifying individual cells and doing all of these morphological profiles across each of these stains, which then gives us an extremely deep feature matrix, right? So you end up measuring somewhere around 3,000 features on each individual cell. And then you can cluster the cells and they give you information about sort of like chemical effects and mechanism of action. So this is just like a cute little picture that shows you about how each of these stains look under the microscope. So you have like the 5 different channels that are capturing information on different organelles in the cell. So very exciting approach, very high throughput, but also very big data, right? And so what's neat about this from the toxicology or EHS standpoint is that chemical exposures alter these cellular profiles or these morphological fingerprints in ways that are consistent with the chemical mechanisms of action. Right, and so here's the DNA channel. If you throw in DNA damaging agents, surprise, surprise, like the nucleus gets messed up. If we have mitochondrial agent, wow, yeah, we get like see alterations in mitochondria, right? So this gives us an idea that the morphological changes reflect chemical mechanism of action, right? And so we wanted to take this forward and do our own study. And so we like got this assay up and running in the lab, which is like not trivial, but Aniga did an amazing job getting this to work. She's like, incredible at this now, like definitely the expert. And so this is part of our breast cancer disparities work where we had identified many chemicals in which there's chemical exposure disparities in the US. And we wanted to assess the effects of these chemicals where we have documented exposure disparities, see like, could they be impacting breast cancer associated biology? So we did many of these high throughput experiments where we're testing multiple doses of these chemicals. And then we also tested a panel of small molecules where we kind of like understood their mechanism of action. That would give us like an anchor to like do like some of this morphological fingerprint comparison. We stained it with the cell painter reagents. We tossed in this like funny microscope we have in our lab that just looks like a big plastic box, which takes like thousands and thousands of pictures. And then we go through this same pipeline where we use Cell Profiler to then do all of the morphological measurements on a single cell level. And then we can go down and import these data into R. We can tell which features are different by which treatment. And then there's a software package from EHS called BMD Express that allows us to start to do dose response assessment process. We can see like, hey, what features are changing at which dose? And then we can see like, are those doses human relevant? So this is like a very powerful tool. Again, why I like this thing costs 25 cents a well, which is like way cheaper than doing a single cell experiment, which costs like $1,500 a sample. So I like it. It's pretty cool. And so this is just like an example of what these data look like. Here's some cute pictures on the side, comparing control versus 2 different doses of a fungicide. Thyram, you can see like the yellow stain there, the yellow punctate stain is the mitochondria. So the mitochondria are like really lighting up. And then we can get these like fingerprint matrices here, right? So here, so like cream is no change, red is up, blue is down. He's like, oh yeah, different chemicals have different fingerprints. This is a simple fingerprint, just comparing the size and the shape of the cells. I would see like, oh yeah, cadmium seems to be increasing most of these. Some other chemicals don't really have an effect. But Nick, who is a very ambitious and excellent undergraduate in the lab, was like, what about all of those features, right? So we had 3,000 features. We'd measured over 1 million cells. Let's see what these data look like. And so he made this fingerprint sort of like heat map now where we can start to see like the similarities and differences in fingerprints. And so it was pretty cool. We had tossed in a bunch of epigenetic targeting small molecules, things like HDAC inhibitors, things that are inhibiting things like DNA methylation. And like, oh, those like all have very similar fingerprints. So like that gives us like some proof of principle. We also see that, you know, heavy metals tend to cluster on the right. For those of you all that can't see, this is like copper, lead, arsenic, and mercury. But what Anaga was interested in is she saw this relationship between DDE, which is a metabolite of the organochlorine pesticide DDT, which is unfortunately still in our bodies, and this small molecule called CHEER 99021, which is an activator of Wnt signaling, which is a really important stemness and cell proliferation marker. So we're then able to dive in on this a little bit more and do an assay where we look at another high content imaging assay where we look at DDE's effects on beta-catenin translocation. So beta-catenin transcription factor, as Wnt signaling is activated, translocates to the nucleus. So in control, we have a little bit hard to see here, but you'll have to take my word for it. Not a lot of beta-catenin in the nucleus. We can then do our positive control, a lot of beta-catenin in the nucleus. Alarmingly, 25 nanomolar DDE also had a lot of beta-catenin in the nucleus. We can quantify this and see like, oh yeah, low dose, very low dose DDE is also activating this Wnt pathway. So we can like validate our fingerprint data now with this orthogonal measurement to give us an idea about, yeah, like here's like a mechanism by which this chemical could be potentially perturbing breast cancer risk. And so I'm going to wrap up by telling you about 2 additional technologies you might want to know about for whole sort of like omics and spatial measurements, which I think are relevant to EHS. And I'll break these into sequencing and in situ hybridization-based methods. And so sequencing-based method, I'm going to compare 2 different 10x technologies here. So the 10x Genomics Visium is a sequencing-based method. You have a slide with individual spots on it. Each of these individual spots has its own barcode. Then you can layer your tissue on top of that and then do a lysis, an amplification, and the barcode from each of those spots gets incorporated into the cDNA pool. And then when you sequence it, you can tell which spot individual RNAs came from. So this is like pretty cool. You're getting whole transcriptome data on around 5,000 spots. Each of these spots is about 55 microns in size. Each of these capture areas is not big, like 6.5 millimeters by 6.5 millimeters. So it's going to be a long time before we can profile like a human brain, Stephen. And these data are discontinuous because these have these individual spots, but these do have the potential to probe different omics layers. So potentially RNA, but also potentially say like ATAC-seq, maybe DNA methylation. So very cool. There's also in situ hybridization-based methods. The method that we just got in at the AGC is called the 10x Genomics Xenium. And so this is a method where we take tissue slices We then incubate them with probes. Usually we're targeting somewhere between 200 and 300 different RNAs at this time. So this is like more hypothesis-driven profiling. You do a round of probe hybridization and rolling cycle amplification, and then you take it to the xenium, and then you're going to do multiple rounds of fluorescent in situ hybridization to identify where each specific RNA molecule lives on a tissue slice. And so you're like cycling through, you find out where each RNA lives. This is very powerful, right? You can see subcellular resolution of RNA expression. Targeted, so like the capture area here is a little bit bigger, subcellular resolution. You still have this like segmentation issue. You have to figure out like where cells are. So this is where those like concepts I was talking about in high content imaging come back. This is still like a microscopy based approach. So you have to be able to like identify cells to tell which cells are expressing which RNAs. And then something that people haven't talked a ton about is that acquisition can take a really long time. Like one of these experiments can take a week of like probe hybridization, washing and removal, hybridization, washing and removal. So it's going to be a while before this is applicable to like a large scale toxicology or human study, but a really powerful discovery tool. And so I think I'm running a little bit low on time, so I might just hit this slide and I'll skip my example. I'll be able to tell you all about it in a little bit, but these are the sorts of things you can do. So we start with intact tissue. Here's a brain sample that's being probed on the Visium. You can then look at the individual spots, do these sorts of clustering analyses. But there's a lot of like neat stuff you can do with these types of data, right? You can do some sort of spatial reconstruction of the tissue based on the individual cell types, where they are. There's a lot of power, and then we can align these transcriptomic data with the H&E image data to really figure out expression at individual tissue structures, right? We can like intersect these types of data. We can deconvolve each of these spots. I told you each of these spots is a 55 micron spot. We can figure out the cell types that make up that each individual 55 micron spot. Our colleague Yue Zhao in Biosets has done a really nice job of this. We can do cell type specific clustering, and we can even look at cell to cell interactions with these data. So this is like an extremely powerful tool that really hasn't been applied yet in environmental health studies. And then kind of like end on this. So there's things that we need to think about in EHS, right? So we're dealing with modest effect sizes compared to, say, if I'm doing a normal versus tumor comparison in a cancer study. So this is something we really need to think about in the study design. A lot of these techniques also have a limited ability to detect locally expressed genes. So that's also something to think about when you're designing your study and what your hypothesis might be. I alluded to this, but biological replicates are extremely costly, right? I told you about how sample handling can be quite challenging. This has been compounded sort of like tenfold with these spatial methods. So be really good friends with your local pathologist and your local histo tech. Super important. Batch effects end up being quite profound in some of these data, something to keep in mind. And then the data analysis is hard. And so if you're interested in learning more about this, I would recommend checking out our single cell spatial analysis program supported by the University of Michigan Biosciences Initiative. So we're building a community of researchers that want to apply these types of methods to challenges in public health and medicine and engineering. If you are not a member yet, come check us out at singlecellspatialanalysis.umich.edu. I think I'll pause there. I do have like another example if folks want to find me afterwards about how we're using the spatial methods in a toxicology study, the spatial sequencing methods.
And this group has a lot of seminars as well. So
yeah, seminars we have. Yeah, so seminars, we have a very robust member group. We have trainee workshops where like the trainees who are actually like doing all the cool stuff get together and talk. Brainstorm, and then we have pilot grants to get new projects off the ground. So check us out. Yeah, would recommend.
Sequencing versus in situ hybridization. Right, so you went through some of the strengths and weaknesses of each. How do you kind of decide?
Yeah, so I think how 10x is thinking about it now is that the sequencing comes first. That's like your discovery approach because you get the whole transcriptome picture. Once you have an idea about what might be changing, then you can do the targeted in situ hybridization-based approach. So we're like, we haven't tested an in situ hybridization-based approach yet. I think 2 people have used the Xenium so far, and I think one of those runs didn't necessarily work that great. So this is like still quite new. And so we're going to order a panel of Xenium reagents, breast cancer focused. So we'll have an idea about sort of like the strengths and weaknesses. But I think in general, the idea is sequencing first, come up with your gene set, and then take that forward to ISH. Claudia.
How about also including your high content, especially with the home game, because it gives you an idea of which actionable profiles or pathways may be of interest, and then you may be able to get a kind of target some of those pathways. If it's mitochondrial, you can get a couple, and then you can get targeted first profiles also.
Yeah. Some of these panels can also add on protein, right? Which is cool.
Yeah.
So you can do like antibody add-ons. So yeah, so it's like something we've been like trying to brainstorm how the experiment would work. So like do the cell painting staining plus the in situ hybridization. So we have some like ideas, but it's not true. No one's done it before.
No,
So it would be
it's
cool.
cheaper than doing a tenant sequence. I mean, you have an idea of
Yeah.
what you can go.
For sure.
With the high content.
Yeah, it was
But
super
I
cool.
think high content imaging is not done, to be honest.
That's also my feeling. Yeah. Maureen.
This will be a quick question. So is it possible to do the cell painting and the spatial sequencing on the same samples, or?
I don't know. Yeah, that would be cool to test. I think maybe.
That
Yeah.
would be really cool to see the association.
I know, yeah. We've always had a dream that like if we do the cell painting and the spatial profiling, so then you have like the morphological fingerprint for each cell plus the transcriptional fingerprint. Could you like learn the transcriptional changes from the morphological changes and then just like skip doing the transcriptional profiling eventually and just sort of like predict the transcriptome from the cell painting data?
Not sequencing in the world.
Well, then you lose, then you don't know which cell was which, though. It's like,
No, it's not.
yeah, you know what they're called. Yeah, so we've like done it where we like done them in parallel, right? Where we've done like cell painting plus RNA-seq. But like the dream would be like having that info on the same cell. That would be like crazy.
That would be a science paper.
That would be a science paper.
So this cell painting, is it done like in a lab or?
Yeah. Yeah, it's actually like really easy. It's like, it's not easy. I don't want to downplay it. But as far as like things like this go, it's easy. Yeah. And it's very portable from lab to lab. And it's also very portable from cell type to cell type, which is cool. Yeah, happy to chat further.
Does the coordinate, the AGC do that? No, okay. I have to come to you.
Sure. I mean, Johnny Sexton, they got to help us get it right. I don't want to like that. But yeah, we can help get things off the ground.
I have a question about one of your data figures that Nick did with the morphological fingerprint.
Yeah.
That is so cool. I think it's outstanding. What tissue was it? Or like how?
Oh, these are normal breast cells.
Oh,
Not normal,
okay.
non-tumorigenic breast cells.
Okay.
MCF10A something.
And then they were treated with all these different chemicals.
Mm,
That's outstanding. Congratulations. Is it published somewhere?
no. Not yet. Thanks. Bring it on.
Perfectly with the project that we're doing in our lab. So
Mm,
yes,
we should chat.
yes, we should. Yeah.
mhm. Yeah, hopefully we'll get it up on next month or so. Cool.
Okay.
Great.
Thanks, Jeff.
Thanks.
Next, we have only 1 hour for 4 really great early career scientists. So these presentations are just going to be a snapshot of what they do, and we won't have a lot of time for questions, but please stay for lunch and, talk to everybody and network and ask your questions then. So first we have Dr. Yue Zhao, who we get to hear more about ALS again. And so Dr. Zhao is a new research assistant professor here in our bioinformatics department. And she comes here after getting a PhD at University of Central Florida. And she's an expert in multi-omics and is applying that to ALS. So we're very lucky to hear about that today. Thank you.
Hello, everyone. So today I will talk about the DNA methylation age acceleration and consider it as a link between the occupational exposure and ALS patient survival. So special thanks go to Tina Lee, who is a wonderful graduate student, has done the majority of this work. So first, I want to introduce some concepts. So what is chronological age? Is the calendar days passed by after birth. And on the other hand, what is biological age? It depends on the biological states of each individual. So ideally, we wish to measure the biological age of each organ of our human body, like a brain, liver, hearts, or even every tissue or every cells. Since the majority of the cells all have a DNA, so it's natural to think about to use the chemical modification of the DNA molecule as an estimator for the biological age. So this is also called DNA methylation age or epigenetic age. What is really interesting is, or really related to the physiological state of each individual, is the DNA methylation age acceleration. So here on this line shows the population mean or population average of the age acceleration. So if an individual has a higher epigenetic age acceleration above this line, then usually it has more risk to develop certain type of disease, especially the aging-related disease. And as mentioned, or as taught by Stephen, so amyotrophic lateral sclerosis is a very typical age-related disease. The average onset, the average diagnostic age is about 55 years old, and it's like effective treatment, and the cause of this disease is quite complicated, has both genetic factors and environmental factors. So also he talked about the multi-step model. It's relates the disease to gene, genetic background, the time, the aging of a person, and environmental factors. So previous study also showed that the DNA methylation can serve as the epoxy of post-environmental exposure of a person, and also serve as the epoxy of the aging. So in this study, I will show that how this DNA methylation or methylome of ALS patient can be linked to both aging and environmental factors. So here is our study cohort. We have over 200 healthy controls and over 400 ALS patients. So those are demographic match. And I also listed the important clinical variables for the ALS patients. We have a very repeatable bioinformatics workflow. And this is Raymond who has done this bioinformatics analysis. And starting from the epic array data, all the way after the quality control, and we got this beta value and the M values. So beta value is more easy for biological interpretation and for visualization. We use it as an input for the age calculation, biological age calculation. And the M value is good for statistical use. So we use it for the differential methylated CpG probes detection. So here we put the input for the DNA methylation age calculation include both age, sex, and the DNA methylation beta values. We use the latest tool to calculate the DNA methylation. So they have several different generations of DNA methylation. Here we use the GreenAge tool to do the calculation. And then after we get this DNA methylation age, we calculate the average based on the healthy controls. So this green line is the expected methylation age. And if a person has a dot, like the red dot has is above the average expected methylation age, we call it has a fast aging rate. And if it's below this line, it has a slow aging rate or slow age acceleration. So here, let's look at compare the ALS cases versus control. First, we can see the DNA methylation age has a very strong linear correlation with chronological age. And most of the red dots are higher comparing to the blue dots. That is, ALS cases has higher DNA methylation age compared to healthy control. And comparing the age acceleration, the cases has in average 1.8 years higher age acceleration compared to controls. And the next, we look at the age acceleration associated with ALS survival. We stratified the ALS patients into 3 different aging groups: the fast aging group, normal aging group, and the slow aging group. And clearly, the fast aging group has about 0.63 years shorter survival comparing to the normal and the slow aging group. That's corresponding to about 8 months. So that's quite a large number in terms of ALS survival. So this is a quite significant impact. However, there's not much separation between the normal aging and the slow aging group. In order to consider other important clinical covariates, we include the onset segment and the symptom onset age and all the other important clinical variables into this Cox-PH regression model, even accounting for all those important clinical variables. Age acceleration still play a very important role in the ALS survival. The hazard ratio between the fast aging group and the normal aging group is 1.52. And we wonder why it's only a fact, we only observe the significant effect in the fast aging group, the normal and the slow aging group. So we went ahead to look at the correlation between age acceleration and all the other 6 clinical covariates, and found that only ALS functional rating score and the sex are significantly correlated with age acceleration. And as expected, the person has a lower functional score, has a higher age acceleration. But this one is not quite as we expected, that the males in average has a higher age acceleration compared to females. So we go ahead to look at the age acceleration association with ALS survival in male and female separately. And males have very strong correlation or association between the age acceleration and the survival. However, females doesn't show any effect. So that's quite surprising us. And we wonder, is that due to, so why is only significant in males? Is that due to any biological reasoning or environmental reasoning? So first, we look at the environmental factors. As mentioned by Stephen, there are different occupational risk group. We have high risk group and a low risk group. So a subset of this EPIC sample also have this group as the occupational risk group assignments. So we compare the high risk group in male and the low risk group in females in case and control separately. And for these 4 different groups, only male ALS cases are strongly associated with DNA age acceleration. And all the other, like a female case, male control, female control, there is no significant signal detected. So only for male cases, DNA methylation age is strongly correlated with occupational risk group or environmental risk. And let's step back. We show that there is a sex influence on the DNA between the DNA methylation age acceleration and the patient survival. And we look at the environmental factors. And the next, I'm gonna introduce the biological reasoning study we have done. And the first, we'll start from the transposable elements. Why is that? There is a pathology called TDP53 pathology. That is a fact involved in over 95% of ALS patients. So it's quite important. And it's known that the TDP43 protein is a DNA binding protein. It binds a subset of transposable elements, line sign and ERV, and it contributes to the silence of those transposable elements in human genome. And the loss of the TDP43 in ALS patients will cause those transposable elements to be reactive. So we want to study if this is true in our cohort. And to do that, we first overlap the DMC, that is the differential methylated CpG sites, and the TE region. And if it's in the not TE region, the hyper and hypomethylated CpG sites are quite balanced, 50-50%. And in the TE region, it's majority of them is hypomethylated, which is provide evidence for the transcriptome reactivation of those TEs. And this observed in all 4 TE families we studied, DNA transposon line, sign, and LTR. And then we further calculated a TE score based on the beta value for each patient. And then we show that there is a strong correlation between DNA methylation age acceleration and this TE score in the DNA transposon hypo and the line hyper score. But this phenomenon, this correlation are similar observed in both male and female. So this should not be the reasoning for the sex influence we observed, or the sex difference we observed. Next, we look at the transcription level change. I will go through this quick. So we first compare the case versus control, the differential expressed genes between case versus control. And there are over 1,000 BEGs we detected, a very strong signal. On the other hand, when I compare the fast agers versus the slow agers, there is only less than 50 DEGs detected. So the signal is quite weak. But still, overlapping these two give us 5 interesting genes. And the top one is the most interesting one because it's already shown some functional related in the nervous system. We'll continue to look at this gene in particular. So this is still an ongoing study. And the next, we'll look at the cell type composition and the DNA age acceleration. So the age acceleration is strongly associated with an increase in neutrophil percentile in both female and male. And also it's strongly correlated with a decrease in all the other cell types. And this association is slightly stronger in the males for most cell types. So in summary, I showed that the ALS patients have higher DNA methylation age acceleration compared to the healthy control. And the greater DNA methylation age acceleration is linked to a poor survival outcome only among ALS cases, only among ALS males. And we studied the environmental factors, transposable elements region, RNA expression, immune cell composition, and to look at what is the reasoning behind this sex difference. And we conclude that the age acceleration is partially stemming from the environmental exposures and potentially from other male-specific effects, for example, the immune cell composition. So finally, I want to acknowledge my wonderful collaborators and also the funding supports.
Thank you.
Okay.
Very interesting talk. And have you thought about race as an important covariate for DNA methylation age? And if yes, can we incorporate that into your analysis?
Yes, great question. Yeah. Actually, DNA methylation age is really race dependent. Yeah. But since I think our cohort, like over 90% is Caucasian, right? So, we can put it as a covariance, but I think the effect will be the signal is not very strong. Yeah. Patty.
Thank you for a great talk. I really enjoyed that. I was really excited about your neutrophil observation on the second to last slide. Could you speculate on that a little bit? I know there's a lot of interesting hypotheses about immune cell dysregulation in the ALS.
Yes. ALS cases in general have a higher neutrophil percentage comparing to the healthy controls. And also, we see that it's, I think it's also has previous study shown that the neutrophil percentile is increasing with the DNA age acceleration increasing. And I think it's the driving force in the immune cell population. Since it's increasing, so all the other cell types are decreasing. Thank you. Okay.
Great talk. So this is a really interesting observation about the sex-specific effects on DNA age acceleration. But I noticed that females in general just have a really poor survival.
Yes.
Do you have any hypotheses there?
That's also, great observation. Yeah, I have been thinking about that. Like, no matter which age acceleration group the female belongs to, it all has the worst, similar survival, similar as the male fast aging group. So I think that shows the complexity of the ALS disease. It's not only genetic background, is also environmental factors, but maybe because we don't have enough female in the high risk occupational group, so we couldn't detect this signal. Go ahead.
So I think it's difficult, and not to talk about any gender-specific stereotypes, but I think sometimes too, there may be a certain effect that's better at caring for other individuals, and so sometimes what we see is that if you need a good caregiver to help you live longer, there may be certain people who are better caregivers than others. Again, not to change gender-specific stereotypes or things like that, but so I don't know, but I think it's a more complex issue than just. I think there may be other factors beyond what data are presented here that may be driving some of the differences that we're seeing. And I want to be cautious with my words in 2023, but I don't know if the data fully teased out here. There's a lot of because I know this is like a short amount of time.
Yeah, but certainly there are some other factors we probably not capture in this study. Yes.
Right.
Yes.
Just curious what the inheritance of ALS is, and whether something
Sorry, excuse me, Caleb.
inheritance, whether something epigenetic factors can pass on next generation.
Oh, okay, great. Yeah, great question. Yeah, we don't have this study, but genetic.
I'll take that. So there's a registry out of in Ireland, and the lifetime population risk is about 0.3%. And for those who have a family history of ALS, that risk increases up to 0.7%. So that gives us some idea. But the other thing that's really interesting is that the higher risk groups tend to actually be the mother to daughter pairs versus the other combination. And I don't think anyone really knows why that is. So even within those groups, a mother to daughter risk is higher than, say, mother to son or father to son, father to daughter. But there are a lot of the sex-specific effects in ALS are quite incredible. We see it across we see it across our immune analyses. We see it over and over again. And it's something that we're really, really interested in. My colleague, Ben Murdoch, gives this talk, ALS, sex, and demographics, drug, sex, and rock and roll. I think so like drug, sex, and therapeutics or something, because, we're just seeing so many differences from males to females. And we know that research tends to under capture, to not necessarily capture the patients that we actually see in our clinic, and that tends not to necessarily investigate these biological differences, which we need to do a much better job.
Okay, so next up.
Yeah, so they're all here. We don't need to keep things like that.
Okay, next up we have Dr. Kai Wang, who many of you have probably worked with because he's the most helpful person we ever had in the pod score. But today you actually get to hear about his own line of research, which is just as exciting and interesting and neat. And so go ahead.
Yeah. Thank you, James. Yeah, so today I'm going to talk about the method this project I'm working on. The topic is that gene target prediction for environmental chemicals using this new method called the coupled matrix completion algorithm. As like mentioned before, so the environmental exposures they can affect human health and cause disease, and new chemicals are like created and released to our daily life like every day. So it's like about 2,000 new chemicals are used in our daily life every year in US, almost like 7 new chemicals per day. And so to do like a comprehensive toxicology assessment of all of those new chemicals is impossible because we don't have enough money and time to do this. So there are like computational toxicology methods. So previously, those methods like the QSAR, SAR, real counts, and other machine learning methods like SVM, deep learning, random forest, those kind of methods is very popular in the toxicology research. But usually for those methods, they are only like treat how they works is like they will predict a chemical, whether this chemical can cause cancer or not, or there's any marker genes can be like activated by those chemicals, or the specific pathways can be activated by those chemicals. So it's more like a binary classification problem. So it cannot give like overall biological effects of the chemicals. So now there's another method called matrix completion. So this method is like a very popular in the drug target research. So they are like in the drug target prediction, they are using this method to predicted whether the drug can be like a target to specific proteins or genes. This method is kind of like so we have like a partially observed matrix here, and then we use some mathematical ways to like finding the missing values for those observed matrix. And so for drug target interaction prediction, most of the input matrix is a binary matrix because drugs are designed for target specific genes, right, or proteins. So basically, people use those binary matrix to represent the physical interaction between the genes and drugs. But for environmental chemicals, they're not like designed to target any genes, right? So in this study, so we are not like only focused on the physical interactions. So we are like actually taking all the type of the interactions into the matrix. So here we use this this is a new matrix completion algorithm, and it's called coupled matrix completion. And this method is already used in the drug target prediction. So then we use this method to the environmental chemical interaction prediction. So the input of this method, they have like 3 matrices. So the first one is a chemical gene interaction matrix, the MEG here. So most of the value in this matrix is 0. So that's why we are using like to finding those values. And there are also 2 coupled matrices. It's MEG and MGG. So these 2 coupled matrices is the chemical similarity and the gene similarity matrix. So after this calculation, so we will have a new matrix that contains all those missing values. And so this is the algorithm. And so the data we used for this project So the data for the main matrix like contains the environmental chemicals and the gene targets interactions is from the CTD database. So the database contains those environmental chemical and human gene targets data. And we use the human data from this database. And after we're filtering some drugs, so there are more than like 4,000 chemicals and more than 20,000 genes. And the known chemical gene interaction number is only like 200,000. So there's a lot, but it's still like a tiny portion of the whole matrix. So a lot of the data is missing here. So we don't know how they interact with each other. Yeah, so this is the data we use for the main matrix. And then for the chemical similarities, we use this RDK. This is a Python package. So we use this package to calculate the chemical similarities based on two types of the fingerprints, Morgan fingerprints and topology torsion fingerprints. And those are the distribution of the chemical similarities. Yeah. So you can see for the topology torsion fingerprints, everything is towards to 0. And for the Morgan, it's a little bit like a 2.1 or something. Yeah, that's a P. And for the gene similarity in this study, we use this package called the Go Simpson. So this is our package that is like measuring the gene similarity based on the Go terms those genes assigned to. So because there are 3 types of Go terms, so we have 3 type of gene similarities: Go BP, Go MF, and Go CC. And yeah, so then to compare our method to other similar methods, so we choose those 3 methods, which are called the CMF, collaborative matrix valorization, GRMF, WGRMF. So those 3 methods, it's all of them are developed for drug target prediction. So it's similar to our method. And we choose those 3 because they also require the main matrix and the 2 coupled matrices. So the input data is the same. But the different part is like for our method, so the 2 coupled matrix is actually directly used into the calculation. But for other 3 methods, it's like they use the 2 coupled matrices to normalize the main matrix and then to do the matrix factorization there. And besides that, there's also a method called the WKN. So we did a key nearest known neighbors. So this method is like a pre-process method to pre-process those 3 matrices. So they can kind of like improve the performance of other those 3 methods. And also for those 3 methods, for every like a matrix completion method is implemented in MATLAB because MATLAB is designed for matrix calculation. Yeah, so but for our method, I implemented into a C++. So I do improve a lot of the speed for calculation. Yeah, so for this testing, we actually generate 3 different sizes of the main matrix. So 200 by 400, 400 by 600, 600 by 800. And our method only took like several seconds for one calculation. So it's really fast. And so we generate the same data set to test all of them. And for other 3 methods, they will run like a with and without the WKNK. And then we calculate the ROC curves and to see how they perform. So here is a result. So the red line is our CMMC method. And so the first 3, the top 3 figure is without the WKNK for the other 3 other methods. And then the lower one is like with the WKNK. So from this figure, you can see for our method, the AUC is a little bit, it's kind of like a stable across the different sides of the matrices, but only drops a little bit like a 0.001 in the 600 by 800. But for other 3 methods, so with a bigger matrix, so the performance like drops like a lot. And also the WKN can improve the performance of other methods. And after this one, so so the ultimate goal of this project is to predict the target gene of a novel chemical that without any biological targets. So to mimic this scenario, so we choose BPA. So why we choose BPA? Because it's very well studied. So we can kind of easy to validate our result. So and also other 3 like chemicals with very similar structure to BPA. To do this is like we will generate a specific data for each of the chemicals. So to do this is like we have the whole matrix, and then we just for each of the case study chemical, we just remove all the existing the interaction values, and then to recalculate those values, then to see which genes we can predict. And actually to run this is like we have, so the total data we have is like we select 10,000 genes, 5,000 chemicals, and contains those, each of the case study chemicals. And then we generate 200 different data sets with randomly select the chemicals and gene from the total gene list. And each of them contains the case study case study chemical. And then we run our method, and then we got all the results. And then we just average everything together to have a total gene list. So for each chemical, the final result will be a list of the 10,000 genes. So ranked by the value we calculated. In this way, so each of the chemical gene pair will be imputed like 10 times average. And yeah, so this is the result. So to validate our result, actually, we are using the data from the EPA Comtox chemical dashboard. So this database, it contains data from bioassays. But for CTD, it's like a literary-based data, yeah. So in here, in the Comtox database, there are 7 genes. That is the common target gene of those 4 chemicals. And in our result, we found 6 of them as a common target gene. The one we didn't find is like, because when I generate data, I don't know the 7 genes. So that the last gene is not in that 10,000 gene. So that's not predicted as a common gene, yeah. So yeah, so this is the result. And there's another thing is like, so this gene is called a GPER1. So this is a target gene. It's only, so our method predicts this gene as a top interaction gene, only for the BPB. And then there's a publication that they found this BPB actually have a much higher 9-fold binding affinity than the BPA to this specific gene. So it's kind of like a support our prediction result. And yeah, so for future plan of this project, for now everything is in matrix, so 3 matrix, the input is 3 matrix. So the next step is like we can increase the 2 coupled matrices to tensors. The tensor is like a 3-dimensional matrix, so we can use all of those different types of similarities then to do the calculation. It's called a coupled tensor matrix completion. So after that, we can expand this method to a coupled tensor completion. So the main matrix can also be a tensor. So the additional diagonal here added here can be like different tissues or different like dose reactions here. Yeah, so this is a future plan for this project. So yes, and I want to thank my my great advisor, Maureen, Dina, and Justin, and all other people on this project. And we also submitted a K99 grant, and I got an iPad score of 15. So really amazing. Thank you. Yeah.
So nice program. I kind of impressed that your method outperforms other methods in AUC and faster than those. So do you have can you explain how you could make a better, performances compared to other methods?
You mean the
in AUC?
AUC? Oh, yeah. I think one thing is like for all the drug target interactions, they are using a binary matrix as an input. But for our data, it's not binary. So it's like all decimal numbers. I think that is probably one of the reasons. Another reason is like for other, as I mentioned, that those other methods, they are only use a couple of matrices to normalize the main matrix. Then the calculation actually is only based on the interaction matrix. But for our method, is like we will grab the corresponding information from the couple of matrices to do the calculation. Yeah, that's why we can have better performance.
I see. Thank you.
Yeah.
One thing that's usually missing, especially for me in the study, is that we're rarely exposed to one compound at a time. So by tapping all of this resilience of information, are you going to be able to try to decipher interaction? It would be very interesting to start to see how different compounds interact together? Because we're always a sporting relation.
Yeah, that's a great question. Actually, for now, for this project, we are not like actually.
I'm not talking this way.
Yeah, for next one we will. Because the thing is like to define the chemical similarity of the mixture is kind of like a tricky part. So yeah, that would be in future plan. Okay.
Okay, so next up we have Dr. Rachel Morgan, who just graduated from our EHS PhD program and is now working as a postdoc with Justin. And so she had a really ambitious dissertation that included cells and mice and humans. And so I think today we just get to hear about one small part of that.
Well, Justin already talked about the cells, so.
Yeah, you saw the cells
We got a little
earlier.
bit about-
So here's another part of that project.
Thanks, Jackie. All right, I'm gonna try and stay on time because I know we're running over. But yes, as Jackie mentioned, this is one component of my dissertation, which I defended a couple of months ago. So apologies to those of you in the room who now are hearing this twice in one summer. But this is a project that I have been working on with Dr. Kai Wang, where we're looking at changes in DNA methylation at imprinting control regions following perinatal lead exposure. And so we're going to highlight some of the sex and tissue specific effects that we saw and some maybe implications that this might have for some future work moving forward. So in the research groups that I've been involved in for the last 5 years, both the Dolino and Colacino labs, we're really interested in how developmental exposures are potentially associated with the onset of disease in adulthood. And so one avenue by which this relationship is thought to occur is, of course, epigenetic mechanisms, which we've talked about a bit today. And just as kind of a primer for everybody in the room, of course, when we talk about the epigenome, we're talking about chemical modifications to DNA and histone proteins that regulate gene expression without changing that underlying DNA sequence. And so the mechanism that I'm particularly interested in for the purposes of this study is DNA methylation. And so this refers to a methyl group that is added to a cytosine that is adjacent to a guanine, otherwise known as a CpG base, or CpG site, excuse me. And in general, higher fraction methylation is associated with gene repression, whereas lower fraction DNA methylation is associated with gene activation. So we kind of have this inverse relationship. And this is particularly true when we're talking about regulatory regions such as promoters and enhancers, and one other regulatory region that I'll talk about in a few slides. So it's been shown over and over kind of at this point that environmental factors do alter DNA methylation in specific tissues. And so when we're talking about these environmental studies, a region of DNA that has different levels of methylation between samples is referred to as a differentially methylated region. And so differential methylation can happen in two directions. We have hypermethylation, of course, where we see increased methylation in our sample relative to control, and then hypomethylation, where we see less methylation. And so, as I mentioned already, many studies have looked at the effects of toxin exposure on DNA methylation in specific tissues, but far fewer have actually examined this relationship in multiple tissues from the same individual. And this is a really interesting question and research paradigm moving forward, because it might really help inform some of our epidemiological work. And so this is where the TaRGET2 Consortium comes in. So the overall objective of this consortium, of which multiple researchers here at the University of Michigan are involved, as well as across the country, is to explore whether there is conservation of perturbations to epigenomic marks following environmental exposures across two different types of tissues. So we have our target tissues, which are by and large the tissues where we're interested in the disease endpoints. But as we talked about a couple talks ago, many of these tissues, such as the brain, are really difficult to access. And we almost never get to really appreciate them in their full glory in our epidemiological and clinical studies, unless patients or participants have passed away. So we have an additional tissue category called surrogate tissues and cells, where we're looking at things like blood samples that are much more readily accessible in our epidemiological work. And so we're interested to see whether or not changes in these epigenetic patterns are conserved across these two tissue types.
And so we're using mouse models to really explore this question using environmentally relevant doses of a wide variety of exposures. But for today, I will be talking about kind of cortical and blood tissue, and then lead as our toxicant of interest. And so the study paradigm that we employed involved exposing the F0 generation of dams to 32 parts per million of lead beginning 2 weeks prior to mating, and then exposure lasted the duration of gestation and weaning for that F1 offspring generation. So we really had this exposure that lasted through two really critical periods of development. Upon weaning, this exposure was terminated, and these mice were allowed to grow to 5 months of age, at which point they were sacrificed and we collected blood and cortical tissue. From there, we performed genomic extractions, and we used this DNA fraction for DNA methylation analysis via WGBS. And so WGBS stands for whole genome bisulfite sequencing. This is a wonderful and very thorough way to assess DNA methylation. And it works by leveraging a technique called bisulfite treatment. So under bisulfite treatment, unmethylated cytosines will be converted into a uracil, which upon PCR amplification and sequencing are read as a tyrosine, whereas methylated cytosines are actually protected from this process. And so we can actually look at kind of the relative proportions in our untreated and treated samples to get an assessment of the percentage of methylation at various sites in various regions. And so when we look at kind of the broad trends in changes in DNA methylation following this lead exposure, we actually saw some very tissue and sex-specific patterns in terms of DMRs. So beginning with the female samples up here on the screen now, we have it broken up into the cortex and the blood. We saw the overwhelming number of differentially methylated regions appeared in the cortex, followed by the blood, and a really small percentage actually overlapping between the two. And this pattern was replicated in the males as well. Again, the overwhelming number appearing in the cortex, followed by the blood, and pretty few actually overlapping between the two tissue types. When we look at overlap between the sexes, we again see very little overlap. So in both the cortex and the blood tissue samples, we only had roughly a dozen, give or take a few, DMRs that overlap between males and females. And those of you who work in toxicoepigenomic research at large are very well aware that sex usually is a very big factor in our results. And so this kind of reestablishes our concern for sex when we interpret our results. Bless you. We mapped these DMRs to genetic locations and then conducted a pathway analysis, which revealed, again, very unique profiles in terms of the biological processes that were being affected by these differentially methylated regions following exposure. One commonality, actually the only commonality that we saw though across these sex and tissue combinations was genetic imprinting. So we saw that methylation changes were detected at genes involved in genetic imprinting in both tissue types. And the regulation of imprinted genes actually relies very heavily on epigenetic mechanisms. So this was interesting to us as we continue to explore our results. And so just to take a moment to reflect on imprinted genes a little bit more, non-imprinted genes represent genes where you inherit a copy of each gene or an allele of each gene from both paternal and maternal genomes. And they both kind of have this equal chance of being expressed in a given cell type. However, imprinted genes are an example of tissue-specific monoallelic expression. What we mean by this is that only one allele is expressed, and it's very tissue dependent and oftentimes developmental period dependent on which allele is expressed when. And this expression pattern is determined in a parent of origin manner. So what this means is we see the allele that is expressed is determined by differential methylation in the promoter region or what's called the imprinting control region. So if the maternal allele is imprinted, we see this increased level of methylation on that allele, and we'll actually see the paternal allele expressed, and then the inverse in the case of paternal imprinting. This is also really interesting when we think about developmental exposures because imprinted genes are known to be pretty important to things like growth and early development. So imprinting control regions really just represent genetic elements that confer imprinted allelic expression to more than one gene. And so when we think about our toxico-epigenomic studies, this is particularly interesting because any exposure that impacts an ICR could potentially be impacting more than one gene at a time. And so we could potentially be seeing this kind of magnitude of results as opposed to this one exposure, one epigenetic change, one gene affected paradigm that we're used to. And so I'm skipping a few steps here in the hopes of being brief, but we did see several imprinted genes that were affected by this exposure in terms of their differential methylation. And then we honed in on two in particular. So these are GNAS and GREB10. GNAS itself has two ICRs represented by these brackets, whereas GREB10 has one that we focused on in this study. And so when we looked at regions of differential methylation, we found that, we had kind of two different examples of our results presented here. So in the case of GNAS, we saw some DNRs that appeared in these ICRs, but also in other regions of this locus. Whereas in the case of GREB10, we saw the vast majority of ICRs actually, or vast majority of DMRs actually localized within that ICR. And when we did some statistical testing, we actually saw an enrichment for DMRs within those control regions. What was also very interesting to us is that we actually are starting to see, or at least have the inklings of, a replication of an epigenetic signature following this exposure that's being found in both a target and a surrogate tissue. Now, this is very sex-specific. We only saw this in males. But we did see that there are DMRs in the GREB10 ICR in both the male cortex and the male blood, and that they are happening in the same direction. So we're seeing them both kind of hypermethylated in this case. However, we didn't really see anything in terms of the female samples and nothing very robust in the GNAS locus. I do want to highlight just some limitations of this work because it does inform kind of some next steps and maybe research interest moving forward. This, of course, was a bulk tissue analysis, and thanks to Dr. Colacino, we know that there's a lot to be gained from cell type specific analyses moving forward. We were also unable to assess allele-specific changes in DNA methylation, which is really important for imprinted genes. However, because this was kind of a discovery approach where we kind of let the results tell us where to go next, we weren't able and aren't able to kind of go back and do that analysis or those types of assays ad hoc. It would require an additional study moving forward. But in general, this study really gave us some pretty exciting conclusions. One, by and large, the effects of perinatal lead exposure on DNA methylation are detectable into adulthood. Many changes do appear to be sex and tissue specific, but when we're talking about this TaRGET2 Consortium and that objective, imprinting, control regions could be loci of potential when searching for biomarker effects in the brain when it comes to this particular exposure. I do want to acknowledge by and large my dissertation committee, most of whom are here, as well as Claudia, who really led the WGBS itself, and Wang, of course, who's helping me write this manuscript. I will also highlight that this manuscript is much larger than what I'm presenting today. We have a whole nother tissue, including the liver, and a whole nother exposure, being DEHP. So if you're interested in that, please keep an eye out. It is hopefully coming out very soon. So thank you.
So you mentioned this is the bulk tissue analysis, so you were not able to do any cell type specific analysis. But we have been using the algorithm called BMC. So it's based on the bulk type DNA methylation data. I'm not sure if this can be applied to your analysis or not, but maybe you might take a look.
Oh yeah, we'll definitely take note of that and maybe chat about a future direction. Thank you.
Okay.
Yes, we won't hear last week, but please do talk to all of our speakers at lunch too, and email everyone on their work. Thank you, Rachel. Our last speaker for today is Dr. Pini Perera, a research assistant professor in environmental health sciences. She's an imprinted gene expert that came from LSU over to University of Michigan to apply that expertise to toxicology. And now she's also an emerging leader in piRNA biology. So we get to hear a little bit about what that is today, in case you don't know what piRNA is.
Thanks, Jackie, for the introduction. Thanks for having me here today. I will switch gears and talk about something a little different called PIWI-interacting RNA. We heard about epigenetics. We heard about how environmental exposures impact the epigenome for long-term health. And we also, in the previous talk from Dr. Morgan, we heard that there's DNA methylation, histone modifications that play a role in epigenetics, but I will switch gears and talk about non-coding RNA that also change gene expression without altering the underlying DNA sequence. So for today's talk, I will give you some background information about PIWI-interacting RNA. We call them piRNA. And the exposure of interest for this talk would be lead exposure. We have heard throughout the day, so I can skip through a few slides to save time. And the majority of this talk, I will focus on introducing what piRNA is. They're highly expressed from germline tissues. So how do we tackle this information that we know from one specific tissue type? And how can we apply that to all the other somatic tissues that is interesting to us? And I'll also highlight a database that we generated on piRNA using a method that we developed in the lab. And also conclude by talking about an application, lead exposure effects on PIWI-interacting RNA, and to show that this is sensitive and that it could be sex and tissue specific. So without further ado, PIWI-interacting RNA, piRNA, are a class of small non-coding RNA. So we've heard in the literature that small non-coding RNAs are sensitive to environmental exposures. Things like microRNA is very well studied. And these are oftentimes considered as biomarkers for disease outcomes or for toxin exposures. And for today's talk, I'll be focusing on small non-coding RNA called piRNA. They are very similar in size to all these other small non-coding RNAs, but they're different in certain aspects, and I'll talk about that a little bit in a couple of slides later. So initially, these PIWI-interacting RNAs were discovered from fruit flies when a lot of biological studies were done to figure out what the P element is. So what do biologists do? They mutate or knock down the P element to figure out its function, right? So during those experiments, they found that there are PIWI P element induced wimpy testes. These flies had wimpy testicles. That's how they got the name, fun names. PIWI-interacting RNA had two main moving parts. One is piRNA, the single-stranded RNA itself. They are oftentimes found in clusters throughout the genome and very highly conserved. One important point is that it was discovered in testes, importance for spermatogenesis, and mostly they were found in spermatids. They associate with PIWI-interacting proteins. So two moving parts is the RNA and the proteins that interact with this RNA. The PIWI proteins and the piRNA make a piRNA protein complex. So here it's just a cartoon showing your processed mature piRNAs combining with these PIWI-interacting proteins for its known function. So since this was discovered from germline about 30 to 40 years ago, all the research that followed that kind of followed suit, looking at its function and expression in germline tissues, specifically focusing on testes. So from what we know in testes and germline tissues, these piRNA and PIWI proteins together silence transposable elements. I don't have to introduce that since we already heard about that in a talk before. So from what I alluded to before, we know a lot about germline piRNA function, but a lot of its function in somatic tissues is still not known. For the past 10, 15 years, there have been new papers that have come out about piRNA expression in the brain, hippocampus, and long-term memory in multiple species. So not much is known about mammalian tissues. But it's important to note that there are piRNA biomarkers associated with multitudes of cancer studies. So that further shows that these RNAs are important and they are functionally important for somatic tissues. So in a nutshell, I'll give you a very broad overview of what these are and what they do. And this is an example from what we know about germline piRNA function. As I said before, in mice, there are 3 different PIWI proteins that associate with piRNA, PIWIL1, 2, and 4. PIWI-like, they also have different names for different species, MeWe, MeLEE, and MeLEE2 for mouse, and HeWe for human counterparts. And there are pre-processed piRNA clusters that are in the genome, and these have to go through what's called piRNA biogenesis, what's called a ping-pong cycle. Primary and secondary processing of these piRNA, immature piRNA, to make them more mature with piRNA characteristics that associate with these proteins to go back into the nucleus to silence transposons. So it recruits DNA methylation as a mechanism to silence transposable elements. So all of this to say that if you have higher piRNA expression, it will recruit more DNA methylation. At the target genes, in this example, it would be transposable elements to repress those transposable elements to keep the genome more stable. With that introduction about piRNA from what we know from the biology, I will skip this slide for the interest of time because we know that lead is bad and it's important to study, right? Another point from this slide is that even though we know biologically piRNA is important, there's functions, there's associations to cancer, but there are not many toxicological studies about these piRNAs. The only known mouse study that's there for piRNA associated exposure is DTT and vincristine. So hopefully this year we'll have a paper out on piRNA association to lead exposure. So now I'm getting into more the fruit of this talk is how do we tackle this issue about the tissue, right? And then also how that also leads to an issue with the databases, publicly available databases for our exposure studies and a lot of the small non-coding RNA sequencing. We need a really good database to compare a lot of the big data to get any meaningful information out from that. The bottom line is we need to have more comprehensive databases that allow us to do robust research, accounting for tissues and sex-specific information. So I told you before that I'll go through what a mature piRNA looks like. These are the characteristics of piRNA. So one of the main characteristics is its length, which tends to be about 24 to 36 nucleotides long, based on what we know about the germline. So a lot of the piRNA research, to detect piRNAs, a lot of the researchers use one or more of these characteristics to call them piRNA, to detect piRNA and generate databases. The next is its preference to a uridine at its 5' position. And typically their 10th position is an adenine. And most importantly, what I'm going to use for this talk is this 2' O methylation modification at its 3' end. And as I mentioned before, they are clustered throughout the genome and highly conserved. So in detecting piRNA, there are several ways to do that. There are already existing databases. Unfortunately, they only have a lot of germline piRNA sequences and germline piRNA information. And it also has multiple methods of piRNA detection pooled into one database. So for example, we could use what's called peridate treatment or peridate oxidation as a chemical experiment to detect or enrich for sequences that have this 2' O methylation modification. So if we can enrich for this modification, sequence that small non-coding RNA, and then look for the size and these other characteristics, we can confidently say that those are piRNA. Another method that's oftentimes used is RNA co-immunoprecipitation methods. So one challenge and a pitfall there is the purity of your antibody. Not all. So since the antibodies may not be from the same batch, and one protein to another is different. So one experiment to another, there could be changes. So the protein and the RNA that you pull down may be different. So from one experiment to another, there's less chance of having scientific reproducibility. So that's why for our study, we use this chemical oxidation method because that kind of makes things more streamlined. So our first goal was to show that PIWI-interacting RNA and piRNA are expressed in somatic tissues. So to orient you to this slide, it's a kind of busy slide. We use this peridate treatment on non-exposed mice and looked at testes, which is our control tissue, and all these other somatic tissues. And on this middle panel, you're looking at frequency on the y-axis, and the x-axis is showing the piRNA length. And it's shown down below. But for ease, all the blue bars represent all the transcripts, the piRNA sequences that are shorter than 20 base pairs, and yellow represents everything that's longer than 20 base pairs. So here in parentheses, you can look at the number of piRNA transcripts that we found in each tissue. So consistent with literature, we found high piRNA expression in testes. And among all the tested somatic tissues, we found high piRNA expression in the brain. So cortex and hippocampus are parts of the brain. So we see that there is high piRNA expression in the brain. And here we're looking at testes. If you look at the length distribution, they tend to be about 24 to 36 base pairs long, one of those characteristics. But if you look at the somatic tissues, they tend to be on the shorter side. So there are piRNAs, somatic piRNAs that are within the 20 base pair length, but not necessarily the 24 base pair length. This is important because a lot of the researchers use size as its sole characteristic to figure out that they are piRNA. So if you are doing a study and then the database is just calling them piRNAs based on just size, 24 to 36 base pairs, you might be losing these smaller piRNAs in your data analysis. So, and this web logo on the very right hand is just to show that the other characteristics, the uridine signature at its first position and the A at its 10th position is still maintained in these somatic piRNAs. And what, thanks to Dr. Kai Wang, we generated our own database so that we can compare our exposure studies, do small RNA sequencing, and then compare those tissues and accounting for tissue and sex-specific differences based on one specific method, which is this peridate treated small non-coding RNA. And this is a busy slide just to say that we have 3 species in this database: mouse, macaque, and humans. And that we have information on like genomic locations, gene annotations, Repeat annotations, because why? PIRNAs are important for transposon silencing, so we need to kind of take a deeper look into that. And also another function that may be associated with somatic tissues is mRNA targets. That there's another function, lesser known function of PIRNAs is gene transcriptional regulation that we oftentimes forget to mention because the main function is transposon silencing. But this allows us to use this database for future research, whether it is a knockout animal model with a disease or a disease itself if we have human data, and also environmental exposure studies with age, tissue, and sex-matched information to analyze our data. So in sum, we know that PIRNA is expressed in somatic tissues, including the brain, and that somatic PIRNAs are shorter in length compared to the germline, and that we have generated these full databases that you can use in the future. We hope to submit this in maybe next week, right, Kai? So stay tuned. So the application. So going back to what we know about the mechanism of this PIWI-PIRNA pathway and its role in the epigenome, our next question is, how does lead impact this mechanism? Is PIRNA sensitive to lead, and is that leading to downstream molecular outcomes? So to answer this question, we used the target animal study, which Dr. Morgan mentioned. So I am not going to spend any time on this. So there's lead and control animals. Just like her, I'm using animals that haven't seen lead for 5 months. They've only seen it in utero during their development. So we used our small RNA isolation from somatic and germline tissues. Using these two different cohorts, we use the brain, the liver, and the gonads. So we have 4 males, 4 females. So you would only have half the gonads for each sex. We size selected, library prepped, and sequenced these small non-coding RNA, and Dr. Kai Wang helped with the bioinformatics analysis to map the PIRNAs that we found to our known database, and then count the PIRNAs that we have generated and see how many PIRNAs are associated with lead exposure, and to see which direction it has increased or decreased. And also, we looked at these gene expression differences, that's target gene expression differences that may be associated with the PIRNA itself. So I'm not showing all the results. You can talk to me during lunch and I can show you multiple slides of the data. The take-home message is that we found for each of these germline and somatic tissues, which is categorized by sex, so males and females, we would, for testes, for example, we found over 3,800 PIRNAs. And among this, 64 of them were differentially expressed with lead exposure. And 20 PIRNAs out of the 64 were upregulated with lead exposure, and 44 of them were downregulated. What's more interesting about this study is that we found 3 specific PIRNAs. All 3 of them were downregulated with lead exposure, but only in the male mouse cortex. So with lead exposure in the male mouse brain, we found that the PIRNA expression of these 3 specific sequences is downregulated, but I'm not showing the data because of the time, but I have several slides to show all the experiments we did to kind of answer this mechanistic model. We found that one of the target genes, which is called tau K3, important for MAPK pathway, has increased expression, but only in the male mouse cortices that were exposed to lead, not females, and not other tissues either. So we were very confident that there is an association and correlation with this pathway. And we also double checked the DNA methylation using the whole genome bisulfite sequencing data that Rachel and Kai generated. And lo and behold, tau K3 DNA methylation was decreased only in male cortex exposed to lead. So we were able to come up with these mechanisms that kind of is consistent with the known biological mechanisms of piRNA, and that these are some applications that we can use in the future, whether it's for biomarker detection or maybe we can use some of these mechanisms to reverse some of the later life health outcomes. So in conclusion, we know that piRNA is also expressed in somatic tissues, and they are very tissue and sex specific, with the example of the male mouse brain, and that they may be involved in DNA methylation directed mechanisms of lead exposure. And this doesn't mean that there are indirect mechanisms that may be in the play. That's also there. We haven't obviously tested that hypothesis, but that's still there. With that, I would like to thank our great and wonderful team. And a lot of this great work cannot be done with a very strong support system. And thank you for your attention.